Hi everyone, we are now moving on to Unit 6. So this has to do with DNA and replication and gene regulation, so turning genes on and off. So we're going to start with the replication part. You're going to need to roughly know the structure of DNA. I don't think you're going to get to have to go into the background of all of the specific experiments in terms of memorization. You might be presented with some data and have to do something with it, but I'll kind of gloss over that a little bit. We're going to talk about how replication takes place, the enzymes involved, and kind of how do prokaryotes do it versus eukaryotes, and the idea of packaging it all up for the later topic of gene expression. So, DNA is a double helix. I gather you all know that by now. The twisted ladder structure. The backbone or the sides of the ladder are composed of alternating sugars and phosphates. So for DNA, that sugar is deoxyribose, and for RNA, it is ribose. The rungs themselves are the 4 nitrogenous bases, A, T, C, G. In RNA, we will learn that there's an additional nitrogenous base called uracil that takes place of thymine, but this is DNA. In terms of pairing rules, A is always going to pair with T with 2 hydrogen bonds, and you can see based on their particular structure, C and T are a singular ring, whereas A and G are larger. They're known as purines for the larger ones and pyrimidines for the smaller ones. But when they join, based on their particular structure, in this case, A and T are a little bit weaker. They only have 2 hydrogen bonds between them. C's and G's always bond together. They're a little bit stronger because they actually have 3 hydrogen bonds instead of 2. So in terms of heating up to break DNA, it's way easier to break A's and T's than it is to do C's and G's. As I mentioned, the rungs of the ladder are held together by hydrogen bonds, and it holds it together kind of like a zipper. So when we're going to do DNA replication, we unzip those hydrogen bonds and re-zip them up back together afterward. An important thing to know too is that DNA is called anti-parallel. So there is some directionality. Always, we're going to talk about things running in a 5 to 3' direction. That's kind of the head to tail way that everything wants to go. So you can see on the left, we have a 5' end, which usually has to do with the specific carbon that they're talking about on the sugar. So we have a 5' phosphate here, and it alternates all the way down, and then we have this little 3' sugar here. So this is the 3rd carbon. There's a 4th carbon, and then the 5th one up here is the one that has the phosphate on it. So DNA is always replicated in the 5 to 3' direction, and so on. The other strand is actually flip-flopped. So you can see that it's turned upside down, so it actually runs in the opposite direction if we're going 5' to 3', and you can see the sugars are kind of upside down. So that's what we mean when we say anti-parallel. They are running in opposite directions. In terms of how DNA is packaged, DNA itself is about 2 nanometers in diameter. So it's really thin, and there's no way that we could fit all of it into a cell without some kind of packaging mechanism. So in order to do this, it's kind of a beads on a string situation. So we have our DNA, and it gets wrapped around these proteins called histones. And that's going to come into play a little bit later on when we talk about gene expression, because you can kind of wrap the DNA either more tightly or loosen around these histones to allow for more or less expression. But that's kind of how we start to wind it up. Kind of like, a giant extension cord. We're starting to wind it around these histones, and then those kind of clump together as well, and eventually that will package itself into chromosomes. So when we actually look at a picture of a chromosome or sister chromatids, it's really DNA wrapped around histones, clumped all together, looped together, and then that's what we get. So it's very, very densely packed. In terms of prokaryote versus eukaryote, prokaryotic DNA is also double-stranded, just like us as eukaryotes, but it's in a circular form. So prokaryotes have a circular DNA kind of floating around in the cytoplasm. They only have one, whereas we as eukaryotes have linear chromosomes. Humans have 23 pairs or 46 linear chromosomes that are hanging out in the nucleus. Normally, when a cell is not dividing, our DNA is in the form of chromatin, which is really like that spaghetti structure of loose DNA wrapped around histones floating around. Whereas in prokaryotes, it's supercoiled, and they do not have histones. Replication of DNA is known to be semi-conservative. So there were a bunch of experiments that led to this point, but the idea is every time DNA replicates, there is a parent strand. So if we have our old, let's say, DNA molecule that's going to be replicated, the hydrogen bonds between bases are broken, they separate, and then a new strand is formed using the old one as a template. And the resulting DNA strands, you end up going from 1 to 2, are half old and then half newly created. So because of that, we say that it is semi-conserved. Here's a picture version of what happens during the DNA replication process. And we're going to kind of go through all of these elements, but just have a rough idea of the overall process, what comes in, what each of the enzymes do in the order. We did some modeling with this in class, but just see if you can kind of remember and keep all the different pieces straight. In terms of the major players, kind of up first we have helicase, which is an enzyme's job is to break the helix. You'll notice that all of these end in ase, so they are all enzymes. The helicase is the one that breaks the helix. It's responsible for breaking those hydrogen bonds and actually opening up and unwinding the DNA at something called the origin of replication, which is where you're going to start replicating. And it kind of forms a little bubble. It's actually going to go in two directions as it moves. Because replication can happen in two opposite directions at the same time. We have something called topoisomerase, which is basically the unwinder. It actually moves ahead of the helicase. So if you think about DNA as a twisted ladder, the topoisomerase really untwists it so that helicase can come in and then unzip. And they are also responsible for kind of putting it back together at the end. They rewind after all is said and done. Primase is the thing that primes or gets it ready for replication. Note that this is an RNA primer. So it comes and lays down a tiny piece of RNA that will then flag the next enzyme to come in. That is DNA polymerase 3. So it kind of comes in and attaches to the RNA primer. It uses that as its little kind of landing zone, and it comes in and it attaches. And the polymerase 3 is really what then marches along and adds the new bases. So it brings in the new A's, T's, C's, and G's as it marches down the DNA in a 5' to 3' direction. There's also something called a lagging strand, which is the opposite strand that needs to grow in an overall 3' to 5' direction, but we don't do that. DNA can only be made in a 5' to 3' direction. So this is where we bring in those short little tiny pieces called Okazaki fragments. So each little fragment is made in the 5' to 3' direction and then kind of glued together in an overall opposite direction. A picture will be helpful. If I kind of go back to the one that we had before, you can see a little bit. Here's the lagging strand. So in this case, we have DNA polymerase 3 down here, creating a strand in the 5 to 3' direction, and that's called an Okazaki fragment. And then there's one upstream of it doing the same thing that will kind of run into the next. Then there's one upstream that will make it and kind of run into the next. So you're still generating things in the 5 to 3' direction. It's just you are overall kind of hopping upstream in the 3 to 5' direction. So once DNA polymerase 3 marches down and does its thing, then RNA polymerase 1 comes in. It swaps out the RNA in the primer for DNA, because in DNA replication, we need to end with only DNA. And then ligase is the thing that kind of comes in and glues it all back together. So it kind of helps to replenish those hydrogen bonds between the two sides of the ladder. So kind of going through that a little bit, helicase unwinds. So it creates these replication bubbles. And on either side, we have something called a replication fork. So they kind of move in opposite directions, and you can have multiple going at the same time in a DNA strand. Primase comes in and adds that little RNA primer, while helicase is marching down. Topoisomerase is first unwinding, helicase is coming in and unzipping, and then primase can actually come in and do its thing. And you can see that the original strand here is going 5 to 3'. Therefore, in the anti-parallel sense, the newly synthesized strand is moving in the 5 to 3' direction in the opposite. Polymerase 3, again, is the one that really comes in and brings in new nucleotides. So it marches along and adds those to the chain. The lagging strand is going to be a little bit different, but you can see the polymerase just kind of slowly marching along, extending the DNA. And snippets are one long continuous piece. It will do one long continuous piece on the leading and short little Okazaki fragments on the lagging. So you can kind of see that here too. And here, I would stop this presentation for a moment and just kind of read through and process exactly what is occurring, where 5 and 3' are, and how it's happening. So take a moment or two to kind of really review this slide. Okay, once it's all said and done, you have to proofread, because repair might need to happen if mistakes were made. So DNA polymerase does have the ability to kind of proofread what it did. There are special enzymes that will come in and say, whoop, you made a mistake, a typo, and kind of swap it out. There are also, if there's a messed up chunk, nucleases can come in and kind of cut out a damaged part, and then polymerase will just kind of copy over the mistakes, and ligase will glue it all back together. So just know that during the repair process, there is kind of a proofreading piece. Mistakes will still happen, but they happen a lot less frequently because there's that kind of quality control in place. And another thing to kind of note is on the ends of our chromosomes, we have something called telomeres, and they're short units that are repeated over and over. They kind of act as a cap. And as our body replicates DNA over and over again, we kind of shave off ends after each replication. So it acts as like a little additional barrier so that the good DNA that we really need that codes for something doesn't get chopped off over time. So there is a telomerase that goes in and kind of adds those. And it's possible that certain cells that are damaged over time or lose these caps at the end can then have drastic consequences, because then you're slowly losing important genes over time. So now that we know how DNA copies itself, we're going to be able to now talk about how we go from that DNA to proteins. Some of the things you're going to need to know, just the idea of turning genes on means that they can then go and make proteins. So we have transcription and translation, and really you just kind of need to know the overall flow of events between the DNA being made and then the eventual protein. So transcription, what occurs between that, and then translation, and then at the end, how mutations might impact this. So gene expression just means that it got turned on. And it used to be thought that it was one gene made one enzyme. But then eventually we've learned that our most accurate idea here is that one gene makes one RNA. And then that RNA molecule can then go and be translated into a polypeptide. Now, as we will learn later, that RNA molecule can actually make many polypeptides depending on how we put it together. So there's something called the central dogma of biology. I don't know why it's called that. A dogma is basically like a law or a general rule. And for us, it's the idea of DNA goes to RNA, goes to protein. So the first arrow is essentially transcription. And for eukaryotes, this happens in the nucleus. And then the second arrow is translation, which occurs in the ribosome, which is the site of protein synthesis, so to speak, and both prokaryotes and eukaryotes have ribosomes. On the left, you'll see a prokaryote. So because they don't have a nucleus, all the action has to kind of happen in the same part. They have their DNA that's in that circular, double-stranded format, and then from that DNA, then it gets turned into mRNA in the cytoplasm, and then there's the ribosome. Whereas for eukaryotes, as I mentioned, transcription is occurring in the nucleus because that's where the DNA lives, and then we get something called pre-mRNA. And there's a lot of processing that's involved for eukaryotes because we just like to be extra. And then it can leave the nucleus and go to the ribosomes for translation. Thinking about overall structure between the two, and I mentioned this previously, DNA is double-stranded, whereas RNA is single-stranded. They're both composed of nucleotides. They both have sugars. DNA has deoxyribose. RNA has ribose. The nucleotides for DNA are A, C, G, and T for thymine, whereas RNA has A, C, G, and U for uracil. DNA is kind of our blueprint for life. RNA, as we'll see, has tons of different roles. These are just a few that are known. So we kind of lump the first 4 together into the most common things. So mRNA is what we're going to kind of focus on, which stands for messenger RNA. And they kind of made a pre-mRNA here too, which is the one that hasn't been edited yet. But that's the one that basically takes the message from DNA and then will eventually code it into amino acids. tRNA stands for transfer RNA. This is the one that works during the process of translation and actually brings in the specific amino acids based on the message in the mRNA. rRNA is a main component of the ribosome itself. So the ribosome is composed of rRNA and proteins, and so that's what that is. And then we've discovered in science a couple of other types of RNA as well. There's snRNA, which stands for small nuclear RNA, and that's part of the RNA processing part. It helps the pre-mRNA go to mRNA in the spliceosome, and we'll learn what that is later. We also have SRP RNA. That's a signal recognition particle, and so it binds to specific peptides. Interference RNA, we will come in contact with later also. It has an ability to essentially turn off the synthesis of particular proteins. And then ribozyme, which is a RNA that can actually act as an enzyme, which is kind of cool. So in terms of the genetic code, we've already kind of learned this, but DNA has the template strand. It's acting as the main blueprint. And then mRNA, similar to DNA, goes in the 5' to 3' direction. And so when it's transcribed, it does the same process, and it creates these codons. So if you read mRNA, you read it in 3-letter words, so to speak. So every 3 letters denotes a word or a codon. And that 3-letter codon will code for a specific and unique amino acid. And there will be some overlap as when we move on, but Each 3-letter code codes for one specific amino acid. And here's the chart that kind of shows that. There's one start codon, so every protein sequence is going to start with an AUG, and that's kind of a flag for the polymerase to know where to go and actually do its magic. There's also 3 stop codons, so if reading along the message, the ribosome itself makes it to one of these, it knows that the protein is done and will stop. And then each other 3-letter code codes for one of the 20 amino acids. There is some redundancy. So as you can see here, AC whatever codes for threonine. And so that's kind of how we have 64 different codon combinations for only 20 different amino acids. That's that idea of redundancy. The reading frame, as I mentioned, is in 3-letter words, and so they kind of have to be in the correct groupings, which is why a mutation can have disastrous effects. If you insert or delete one, you're kind of shifting the overall reading frame. The code is universal, so this is unique, not unique to humans, but works for every life form that we know. So let's kind of jump in. Transcription. Transcription unit is basically just the DNA piece that is going to code for a specific protein and get turned into RNA. There's an RNA polymerase, which is responsible, the enzyme, again, responsible for extending and creating the RNA polymer. It's kind of a multi-purpose tool. When we talked about DNA replication, we had a topoisomerase come in, then a helicase come in, then 2 different types of DNA polymerase, and then the ligase would come and glue it all back up. But here in transcription, the RNA polymerase really does a ton. So it's the thing that comes in and separates DNA. It transcribes the mRNA, similar to previous, it goes in the 5' to 3' direction. It follows all the normal pairing rules, except anytime there would normally be a T in DNA, it would be a U. It knows where to attach by looking for a promoter and then the terminator. So we'll talk about promoters a little bit later, and then it stops at a terminator or one of those stop codons. So for prokaryotes, the RNA polymerase binds directly to the promoter, and that's right upstream. Part of the promoter region is that start point, and then you can see it's just going to come in and bind and then move in the 5' to 3' direction down to transcribe. Eukaryotes is a little bit different. There's the start point, but the RNA polymerase does not necessarily bind right to that promoter. A couple of things come in and bind to what we call the TATA box. That is unique to eukaryotes, and it's inside the promoter a little bit upstream. And a bunch of transcription factors that are regulating how much and when and all of those things come in, and then those regulatory factors bound to the TATA box together bring in the polymerase to start transcription. Then we have elongation. So as I mentioned, the polymerase is just a polymerase. It adds RNA nucleotides in the 5' to 3' direction. The RNA polymerase is doing the untwisting and the rewinding. Again, it's a one-stop shop here. And then termination occurs when it gets to one of those stop codons, and that really signals that everything to kind of eject. The polymerase leaves, the transcript stops. And now for eukaryotes, that's called pre-mRNA. For prokaryotes, that's just it. There's no processing, so it's done. But for eukaryotes, we can do more things to it. So things that eukaryotes like to do. We add a 5' cap, so it's a modified guanine at the front, at the 5' end, and we also add a poly A tail at the 3' end. So these things can help the RNA molecule get out of the nucleus, and then it protects it from any of the enzymes that might be floating around in the cytosol that are normally going to degrade RNA. It helps act as a barrier on both ends, and then it also helps attach to the ribosomes. So it just helps with efficiency and protection. There's also splicing that can occur. In this case, there are coding regions called exons within a gene or a transcript, and there's intron regions, which don't actually become a part of the mature protein. So those need to be cut out. So those non-coding sequences are removed or spliced out. And then those exon or the coding regions actually get joined together. So the mature mRNA has a cap, just exons, and a tail. And that together is going to be what then goes on to make the protein. Here you can kind of see what is happening in that splicing event. It occurs in what we call a spliceosome. So this is where the small nuclear RNAs come in. They're called SNRPs. And as long as you know that they just cut out introns, I think you're fine. So why do we even have introns? It's thought that they help to regulate the gene activity. So again, how much, when, those types of things. And because of this idea of splicing, that's how one mRNA molecule can actually end up generating lots of different eventual polypeptides. So we talked previously about how DNA has about 20,000 genes, but we have over 100 different thousand proteins that can be made. So this alternate splicing, where one gene and then one transcript can actually be grouped together in different ways, can yield different proteins at the end. Which now brings us to translation. Translation brings in the message. tRNA is kind of the interpreter. It is the RNA that has an anticodon on one side that's complementary to the mRNA's codon. So that's kind of how that lock and key fits in. And then it drops off the amino acid that corresponds with that code. tRNAs come from the nucleus. They are very specific to each amino acid. As I mentioned just a moment ago, anticodons are on one end and they are complementary to the codon on mRNA. Now, it's interesting too because there's also kind of a wobble as can be seen with the redundancy aspect. So there's 64 codons, but only 20 amino acids. So a lot of times the base pairing that occurs between that last letter and an anticodon isn't super strict, because oftentimes if you have AA something, not mattering what, it'll always code for the same amino acid. All right, the ribosome itself is made up of rRNA, and it has two subunits, rRNA and proteins. It's made in the nucleolus, which is the nucleus of the nucleus, and has a big subunit and a small subunit. And within it, there's kind of this site of protein translation where there's three slots, the A, P, and E region, and we'll talk about those in a minute. So A is where the amino acids are initiated, or that's where they kind of come in. That's where the tRNA's first stop is. Then the P site is for the growing polypeptide chain. So that's where the polypeptide grows larger. And then afterward, there's the E or exit position, where the tRNA can then leave after it's dropped off its amino acid and then go and be recycled, because these are recycled. So the first thing that happens is initiation. And the first codon that always gets read is methionine. So methionine is the first start amino acid to know things that are going to start and that there's a protein coming. So it kind of skips over the A site and goes straight to the P site because it's the first stop in the polypeptide chain. And that brings in the full complete portion of the ribosome to now go ahead and do translation. Before, it was just kind of the mRNA feeding into the small subunit. Now the addition of methionine brings in and completes the set. So the elongation piece is this repetitive cycle. There's codon recognition. So the tRNA that understands that next three-letter code comes into the A site. Then there's a peptide bond formed between the current chain and the new amino acid that comes in and kind of shifts over. That allows the tRNA in the P site to shift over to the E site. The one that just took on the chain is going to shift over to the P site. So that's kind of along our translocation. And then the tRNA will then leave and exit, and the next tRNA will come in to the A site. And then you repeat over and over and over again until your chain is complete when you've reached a termination codon. So there's 3 stop codons that could be reached, and then what happens is instead of a tRNA coming in, a release factor comes in. And then that halts everything, makes the polymerase leave, the parts of the ribosome dissociate, and then leave. It's important to note that there's something called polyribosomes, so there could be many ribosomes working on a singular mRNA at the same time. Just for creation purposes, it allows you to make a lot in a short amount of time. Protein folding is also something that's needed to be considered. Once a protein is made in that long chain, it then needs to oftentimes fold into its 3D structure. It usually does this spontaneously, but there are proteins like chaperonins, it's chaperones or carries, that helps to ensure that polypeptides fold correctly. If the protein doesn't fold correctly, then it oftentimes can't do its job. Before we mentioned ribosomes, this site of protein synthesis can occur in the free ribosomes that are just floating around. These are for things to stay in the cytosol and for the cell to function itself. Whereas again, the bound ones, which are in eukaryotes, are mostly for export. And they can include signal peptides to kind of target where they need to go. If you think about the mRNA having some of those signaling molecules in the head and in the tail, it really tells the mRNA where to go to actually be translated. So mutations can affect all of these things. There's large-scale mutations, which are chromosomal things. So we get these from non-disjunction when chromosomes fail to separate, translocation when a chunk of one floats over to another. Segments can invert or flip-flop. There can be duplications, deletions. That's the big stuff. Point mutations are just one letter. So there can be a substitution. So you replace one letter for another. And as a result, that could potentially mean that now you're swapping out one amino acid for another. That's called a missense mutation. You've misspelled it, so now you have a different amino acid. It could be a nonsense, which means you've accidentally changed the letter, so now the codon codes for a stop codon instead of a needed amino acid. So that halts production of that protein. There could, I guess also here too is important to note, it doesn't say here, there's silent mutations, which that one letter change doesn't confer a difference in amino acid. So it's kind of coding for the same amino acid it would have otherwise. And so it's called silent because you don't really notice the effects. Frame shift mutations are where mRNA is read incorrectly. You can either insert a letter or delete a letter, and because of that, all of the words in that 3-letter code get shifted one way or the other, and now it's all gibberish. Here's an example of a missense. We had AACC before. Now we have AATC. So as a result, instead of glycine, we are now making serine. So that's a missense. A nonsense would be, in this case, TACT has turned into TACA. My goodness. Anyway, now we have a stop codon here instead of coding for lysine, so that causes the premature termination of a message. This is the silent one that I mentioned before, so even though there is a letter change, it still codes for glycine, so you'd never actually see the effect. Insertion, we have added a letter. In this case, the adding of a letter has shifted everything over, so it now causes a stop codon before it should have. It could have also been a deletion. It could have also coded for different amino acids. In this particular example, it causes a stop codon. Deletion, now we're missing a letter. Now it's coding for different amino acids than it would have before. Single letter changes can have drastic effects. One just quick example would be sickle cell anemia. So instead of a GAG, you get a GTG. So it causes a glutamate to turn into a valine. And because of that, they have different properties. If you remember, proteins have, or amino acids have different unique properties. They could be charged or uncharged, polar, nonpolar, acidic, basic. So because of it, it causes a drastic shift in the shape of the protein. And that's why they become sickle shaped instead of the normal circular shaped that red blood cells are. All right, now we are to regulation of gene expression. So how do we control when these genes are turned on, transcribed and translated, and when they're not? So here's just kind of the things that you need to know. For our particular purposes in studying, you hopefully will review the foldable that you had in class. So it was kind of that fold out thing that we made where on one side it had prokaryotes, one side it had eukaryotes. That's a really helpful study tool for this type of thing. Metabolic pathways in prokaryotes are often controlled by positive and negative feedback. So there's multiple steps in one particular pathway controlled by multiple genes or multiple proteins. So you can either control it by the enzyme level, where one particular product might inhibit the enzyme earlier upstream, or you can even have one gene or the presence of one product actually inhibit the gene for an enzyme. So that's kind of the gene expression regulation idea versus just enzyme activity. For bacteria, prokaryotes only, we have operons. They are just a group of clustered genes with an on-off switch. So one promoter can actually control many different genes, and it has what we call an operator, which makes sense for an operon to have an operator. The operator is the on-off switch. So looking at the picture, we have a promoter where the RNA polymerase would come in. There's the operator, which is a part of the promoter that acts as an on-off switch, and then there are particular genes. In this case, there's A, B, and C. Together, all of that makes an operon. So thinking about it, there are also regulatory genes that can act a little bit upstream. So you can see in the promoter region, regulatory genes in a different location can produce repressors that can bind and or control the operator switch. So in this case, we have TRP here that can make, it's a TRP repressor, that when activated, it can actually make a repressor. It's normally not doing anything, but when tryptophan is present, then it can change conformation, come and lock here, and physically block the RNA polymerase from moving forward. When the repressor isn't attached, then this thing can just go. So here's kind of how it works. There's two forms. There's a repressible operon and an inducible operon. So repressible means that it's on all the time until you turn it off. So the polymerase will bind to the promoter and turn on all of these genes normally. There is a repressor, but it's inactive. Something needs to come in and turn this repressor on and then come in. So in this case, there's a co-repressor. When it's present, it will change the conformation of the repressor and it will come and lock in. When it does that, you have essentially repressed this whole thing. It blocks the polymerase from getting to the genes. Inducible is the opposite. It needs to be induced. So it's off until you turn it on. That repressor is locked in. But when an inducer molecule comes in, it can change the conformation of the repressor and cause it to leave. And that will then trigger the transcription of all of the genes that were normally off. So one example of the repressible one, meaning normally on, now we're turning it off, is the Operon. Here's how it works. So the tryptophan, if we have enough tryptophan, we don't want to make more. It's a way to quality control the amount of things that have been made. So tryptophan itself, as a molecule that is made, acts as a co-repressor. So when tryptophan is present, it will bind and activate the repressor that can do its thing and block further production that would lead to more tryptophan. When tryptophan is lowered, then the co-repressor isn't there, and you have kind of the above situation where there's no tryptophan to activate the repressor, so it cannot bind, and therefore you cannot stop the production. Inducible, on the other hand, is usually used only when you're breaking down food. So you're only going to turn it on when you actually need to do something. The lac operon is one particular example. So the repressor is normally there, and then when an inducer binds to it, it removes the repressor so things can actually go. For lac specifically, allolactase can act as an inducer. So when that is present, it will cause transcription of the lac operon, which would then make enzymes necessary to break down lactose. When allolactose is not present, well, then you don't need those enzymes to break it down, so therefore it's not on. These are both, remember, prokaryotes. We've talked about positive and negative feedback before. Negative just means that you switch the operon off by the repressor. In this case, whether you're turning it's inducible or repressible, it's negative control because you're like a thermostat trying to get a happy medium of amount. You don't want to make too much. You don't want to make not enough. So you're oscillating back and forth kind of between on and off. Whereas positive control, as we saw previously, includes things like cyclic AMP, lactase production for pregnant moms and breastfeeding moms, that kind of thing. All right, gene regulation in eukaryotes. So no operons. Eukaryotes are different. We have all of these cells with all of our DNA, but only about 20% of the genes are on at any given time. And different cell types turn on different genes in the first place. So that's how we have a liver cell versus a brain cell versus a heart cell. That is what we call differential gene expression. Different cell types express different genes at different times. And eukaryotes have a ton of ways to regulate gene expression. So again, I refer back to the foldable that you guys have that differentiates prokaryotes from eukaryotes and all the different levels of regulation. So eukaryotes can regulate how tightly wound their DNA is. So we have something called methylation and acetylation that basically winds tighter and loosening DNA to either make it less accessible for transcription and turn it off, or more accessible to transcription and turn it on. So if methyl groups attach to DNA, then it's going to become more tightly packed. It's going to wind around histones a little bit more, and therefore you're going to get less transcription. If you are adding acetyl groups to histones, then it's going to essentially activate. Acetylation, think activation on the histones, then it's going to increase. These are things that are important because they are not in the genetic code themselves. So they're not DNA mutations. This is a piece of epigenetics and can actually be passed down. So that's kind of interesting. The winding tightness is an inheritable trait that's not even in the DNA sequence. Another thing that we can do is activate or repress transcription. So as we were kind of talking about before, there are regulators, activators, and repressors that can control whether or not transcription takes place. It's a lot more complicated than the prokaryotes. Eukaryotes have the ta-ta box. They also have upstream all of the activators and enhancers that might occur. So there's really this folding over business with all of these transcription factors. So all of this needs to happen in order for transcription to take place. So you need to have the right enhancers section, you need to have the right transcription molecules, you need to have the right activators, and all of it together will bring in the polymerase. If you don't have those elements, then you're not turning it on. So you can kind of control the presence of a lot of those things to finally control turning the gene on. It's important to know too that different cell types have different activators. So depending on what activator is available, you're going to get different levels of transcription and in different quantities. So in a liver cell, you might have certain activators that trigger specific genes. In this case, this activator might turn on this gene, whereas a different activator would then cause transcription of a different gene. You can also regulate mRNA. So there are those little interfering and microRNAs that can basically bind to and degrade and or block mRNA from being translated. So even if you do make mRNA and it leaves the nucleus, you can actually stop it before it makes it to the ribosome for translation. This slide, if you want to pause and just kind of read through, there were a bunch of things inside the nucleus with which can be regulated. Again, pause for a moment here too, because these are other elements where regulation can occur. The last piece of this is just biotechnology. I'm not sure that you're going to be asked anything on the AP test related to this, but I just want to go through a couple of things because they might give you an example. Genetic engineering is just manipulating genes and genomes. A lot of it comes in the regards to recombinant DNA. So this is where scientists have been able to artificially make DNA. Usually it's kind of cutting and pasting DNA from multiple sources together. We can also use gene cloning, which basically translates to us utilizing things like bacteria to make a bunch of DNA that we want. Restriction enzymes are scissors. So these are things that cut different spots. So we did an activity at one point where we had a plasmid, and different enzymes have different specific sequences that they recognize and they cut. They have sticky ends a lot of the time, which basically means it cuts in a zigzag versus a blunt cut, which cuts straight. And that's how we utilize that function to cut and paste different chunks of DNA together. Ligase is the glue that is joining DNA fragments together. And we paste it all in what we call a cloning vector, which is a plasmid that has everything that we want. Here's just a chunk of what that might look like. Restriction enzymes come in. We have sticky ends similar that have been cut by the same enzyme on a piece of DNA that we care about. And those little overhangs are complementary, so they will come in. Ligase glues it all together. Voila. We have recombinant DNA, meaning that it's come from multiple sources. We utilize the process called transformation to get bacteria to suck these things up. So we make our special plasmid with cutting and pasting. Then we use bacterial transformation to force the bacteria to suck up the plasmid from the environment. Once it's in there, bacteria have the ability to copy the plasmid independent of their normal DNA, so they can make tons and tons and tons of this like a little factory. Other elements that just might be of note, PCR stands for polymerase chain reaction. That is basically a copy machine for DNA. It amplifies a piece of DNA tons and tons and tons and tons and tons of times in a machine so that scientists can utilize it or use it or test it or whatever they need. Gel electrophoresis is another piece that separates DNA. So sometimes you want to look at different segments. It separates based on size by running electrical current through a box. Here's kind of PCR showing you. This is what a gel electrophoresis might look like. You can load all of your DNA samples in one side. You run a power source or electricity through it, and it separates by size. So big fat molecules get stuck. Small molecules are able to kind of maneuver through. And then you can compare whether it's crime scene to criminals, if it's parent to offspring. There's tons of different uses, but that's kind of just the process. Biotechnology brings a lot of different genetic sources from different species together for the purposes of helping us. So a lot of GMO or genetically modified organisms exist in our food population by trying to utilize good traits to help the food source live longer, last longer, longer shelf life, bigger yield, whatever it may be. At the end here, we have viruses. I don't think you're going to be asked anything about viruses on the AP exam. Just kind of general picture here. Bacteria are prokaryotic. Most of them are free living. Comparatively, they're large, and we use things like antibiotics to kill them. They also are used a ton in biotechnology, like plasmids. Viruses, as we are learning in our current pandemic, are not living. They do infect cells. There are bacteriophages that specifically infect bacteria. We cannot use antibiotics. We can only really use vaccines to prevent and have antiviral treatments. A lot of viruses have a couple main components. They have some kind of nucleic acid in the middle. It could be DNA, it could be RNA, and usually they have some kind of shell on the outside or a viral envelope around it as a form of protection. They do have a lot of different structures, so you can kind of see what those look like. But in the center of all of them is some form of DNA or RNA. They have two ways to reproduce. There is the lytic cycle. You can pause here if you want to go into super detail, but on the left, there's the lytic cycle. This is basically the infectious cycle that is occurring. So a virus infects the cell, injects in its DNA or RNA, and then it circulates. And what ends up happening is the bacteria or whatever the host cell is, that machinery inside that cell, those ribosomes are utilized to crank out tons and tons and tons of virus. It makes all the proteins, the viruses self-assemble until eventually the cell explodes and those little tiny viruses can now infect other cells. So lytic, to lyse something means to break. So lytic in this case can mean the infectious portion. However, sometimes they go dormant, for example. So what happens here is the virus injects its DNA and it kind of goes into the host DNA and just copies every time the cell copies. And it doesn't actually cause the infection at this point, but it can be activated or switch over to the lytic cycle at any point. So that's why there's instances where people can not have a disease for a really long time and then it kind of shows up later. Retroviruses are interesting because they have RNA instead of DNA. So they actually have RNA and they turn it into DNA and then they inject it into cells. So that's kind of one unique example. The HIV is one instance of this. There's tons of different kinds of drugs, as we're currently seeing too, vaccines for prevention and then certain antivirals for treatment. But essentially, you've made it to the end of Gene Regulation, Unit 6.